Hello, ladies and gentlemen. My name is Atisha. Today, my team and I would like to present to you a preliminary study on the influence of temperature, incubation time, and plasma dilution for sensitive EGFR mutation detection via piezoelectric biosensor in lung cancer diagnosis and treatment. Our presentation will include information according to these. Cancer is the second leading cause of death worldwide. Statistically, 1 in 5 people will be diagnosed with cancer at some point in their life. In 2021 alone, cancer was responsible for approximately 10 million deaths globally. The mortality rate from cancer is higher in men, with 1 in 9 men affected compared to 1 in 12 women. One of the most promising emerging techniques in cancer diagnostics is liquid biopsy, which our research aims to implement in future phases. This approach involves analyzing real human blood samples and is gaining popularity due to its non-invasive nature, ability to provide real-time detection, ease of use, and suitability for repeated sampling. At present, the cost of each EGFR mutation test is around 6,000 to 8,000 baht. Ultimately, the goal is to contribute to better treatment outcomes, particularly in the administration of medication. Our study focuses on enhancing the efficiency of lung cancer detection and treatment, specifically targeting advanced stages like stage 3 or 4, which is the reason why we use EGFR biomarker. The doctor thereby can dispense anti-EGFR drugs more effectively. Surosense is optimizing diagnostics by developing a piezoelectric biosensor, focusing on finding the optimal incubation temperatures and time. This forms the basis of our research project. We are very thankful for giving us this opportunity. The heart of this instrument is the QCM chip. This chip has a property called piezoelectricity, which means we can make it vibrate and measure its resonance frequency. The Extens 101 records this resonance frequency and plots it as a graph. What's important is that the frequency goes down as more mass attaches to the surface. In our experiments, we have 4 key phases. First, the pristine state, where the surface is clean and we measure the initial frequency. Second, the probe incubation step, where only the probe is on the surface. And third, when the probe and the target are both bound to the surface. Finally, by comparing the initial and final frequencies, we calculate the frequency shift, which tells us whether EGFR is detected. We have 3 objectives in this experiment. First, we want to find the optimal incubation temperature and time for probe attachments. Second is the optimal temperature and time for probe and target attachments. And lastly is the optimal plasma dilution for the attachments. My hypothesis is that EGFR mutations can be reliably detected using a piezoelectric biosensor because of its high sensitivity and specificity. I also believe that temperature, incubation time, and plasma dilution play major roles in how well the probe and target DNA bind to the biosensor surface, and this directly impacts the overall detection performance.
The following slides shows our experimental procedure. First, we wash the QCM chip with piranha solution, then followed by ultrasonic cleaning with acetone and another ultrasonic cleaning with ethanol. Lastly, we'll use DI water. The duration of each step is 3 minutes. After cleaning, we start the experiment by measuring the frequency of the pristine chip with our equipment Extens 101. Next, we'll add an ssDNA probe to measure the frequency of the chip with the probe, and incubate the chips with a range of temperature and time. Finally, we'll wash the chip with tea buffer, ethanol, and DI water to wash out the unbinded probe. The last step to the experiment is adding target to the chip with probe, then incubating it for a range of temperature and time. After that, we'll wash the chip with tea buffer, ethanol, and water and measure the frequency of the chip with target to plot graph for data analysis. In the result, comparing both incubation for probe in room temperature and 60 degrees, we found that at 120 minutes in 60 degrees, the sensors have a very similar frequency shift, so it can be concluded that 120 minutes is the best time. In this slide, we varied the temperature and it can be seen that at 60 degrees, that the frequency shift are again very similar, so 60 degrees is the best temperature. The probe incubation trials demonstrated a 66% binding rate for EGFR ssDNA probes in 71 trials at 60 Celsius for 120 minutes, reflecting effective probe immobilization on QCM surface. In this graph, you can see that 30 minutes has the lowest frequency shift, which makes it optimum time for probe target incubation. This graph shows that 30 degrees offers the most stable conditions because it also has the lowest frequency shift. For optimum plasma dilution, a dilution of 1 to 200 with T buffer yields the highest frequency shift. High concentrations of plasma results in interference by protein and specific binding, while lower dilution weakens the clarity of the detection. In conclusion, the best probe binding is achieved at 60 degrees for 120 minutes with a 66% success. Hybridization is most efficient at 30 degrees and 30 minutes with largest frequency shift, and the best plasma dilution ratio with T buffer is 1 to 200. Overall, this optimized condition could improve the biosensor detection in terms of reliability for lung cancer biomarker detection.
For future perspectives, we aim to further optimize probe design and hybridization protocols by adjusting parameters such as salt concentration and probe length in order to enhance the sensitivity and specificity of the EGFR mutation detection with our piezoelectric biosensor. Finally, we plan to validate the biosensor using real clinical samples to confirm its diagnostic reliability and clinical relevance. I would like to thank our project advisors and thank you for listening.